Hello everyone. Welcome to the 33rd webinar organized by Manav, the Human Atlas Initiative. My name is Sneha and I will be moderating this session on mitochondrial genomic networks by Dr. Sarika Jalan from IIT Indore. Before we begin, just a note for all listeners, if you have any questions for Dr. Jalan, please use the Q&A box for typing it in. Dr. Sarika Jalan is a professor at Indian Institute of Technology, Indore. Before joining IIT Indore, Sarika was a senior research scientist at NUS Singapore. She has a PhD from Physical Research Laboratory, Ahmedabad, followed by postdoctorate at Max Planck Institute at Leipzig and Dresden, Germany. Her research at IIT Indore is focused on nonlinear dynamics and complex systems with broad research interests in complex biological networks, spectral graph theory, synchronization, machine learning, adaptation, and evolution. She's also the editor-in-chief of the Journal of Computational Science. The talk today will be about mitochondrial genomic networks and how networks have been established as an extremely powerful framework to understand and predict the behavior of many large-scale complex systems. Mitochondrial DNA variations have been acclaimed as one of the key players in understanding the biological mechanisms behind human migration, evolution, and adaptation to extreme conditions. Due to stochastic evolutionary forces, the frequency of polymorphism is affected by changes in the frequency of a nearby polymorphism on the same DNA sample, making them connected in terms of evolution. To explore the cumulative effects and complex behavior of genomic variations in the human mitochondrial genome, Dr. Jalan's group has employed the methodology of co-occurrence and co-mutation networks. These networks reveal that interaction between nucleotides underpin the fundamental processes of human migration and evolution. Applying these networks one step further at a genetic level provided evidence that such co-mutation-based genetic networks could reveal genetic signatures of human adaptation in extreme environments. So over to you, Dr. Jalan. We are looking forward to know more about these networks from you.
Yeah, thank you, Sneha, for kind introduction and Manav for the kind invitation. And let me share my slides. Can you all see my slides? And I'm putting in slideshow mode. And
Yes,
if you have any question or anything, I am also seeing those questions there in the chat box. So feel free to write down your questions in the chat box, and intermittently I will pause and I will take up the questions. So feel free to write down the questions, even though it's a very layman type or whatever, but feel free to write down the questions there in the chat box. So as she introduced, I am from Indian Institute of Technology, Indore. And today my aim is to give you an overview of network field, particularly biological networks. And also to the end of the talk, my aim is that if to the end of the talk, if you want to start working on this network field, you will have enough material to start learning and employing the things. So that is my aim, and feel free to write down any questions during the talk in the chat box. So let me start now. Why this slide is okay? Why it's not working? Okay. So networks, they are everywhere. We live in the world of the networks. Like for example, if you wake up in the morning and look at your mobile, you are using mobile communication network, correct? Then you switch on the induction and then you are using power grid network. You board the bus to go to your office or your institute, and then you're using transport network. Talk to fellow passenger, social network, email, internet network, then browse through web, that is a www. You browse research article if you are a scientist, and then you are using citation network. And there are some review articles which are about, so all these networks have been studied by physicists. So recent last 20 years have seen a spurt in the growth of this very powerful technique, which is the graph theory. So networks are nothing but the graphs, and there are some few review articles which I have written there in order, if you want to explore more about, or in order to appreciate the power of networks in order to understand all these different systems which I have written here. And with this, let me come particularly to biological networks. So biological networks, for example, let me first give you a brief introduction of how a complex system can be represented in terms of network. For example, if I'm talking about an airport, and why I am starting with airport, because social networks or this day-to-day things which we are using is very easy for me to appreciate. So for example, in airport networks or airport system, different airports are the nodes. And if a flight is directly connecting two airports, if there is a flight route between two airports, then we say that those airports are connected. So in this manner, you get a complicated web of these airports and the flight connections. Okay. In www, you have web pages, which are the nodes. And if one web page leads to another web page, then you say there is a connection. And in this manner, you again have a complex system of World Wide Web. In the cellular network, you have chemicals, which are the nodes. And if one chemical is reacting with another chemical, then you can say that there is a link existing between these two chemicals. In the power grid, generators are the nodes, and high voltage links between different generators, they are the networks. And we have a few years back, I, along with my PhD student, we have tried to write down a popular level article on complex networks, and that has appeared into Physics News. So you can get the copy of this one, or any preprint or article. If you want to get later on after this talk, you can mail me and I can provide to you in case you are unable to search them. So now, before I start with this word of network, I have to introduce a bit of technical terms. And then I ensure you that after this, there's no mathematics, no technical term which I am going to introduce. But these technical terms are imperative. So first technical term is degree. And if you intuitively, you will think that degree is simply that how many friends you have. So suppose if you're talking about a social network where you are the nodes, and if you know a person by its first name, his or her first name, then I say that you are friend, for example. Okay. So in that manner, you can define a friendship network, for example, or if you know their parents' name, then you make so you set up the rule, and based on that rule, if a pair of persons, they follow this rule, it means they know each other by first name and they also know their parents' name, then okay, I say that I am establishing a link between this pair of the nodes. So in that manner, you have a friendship network, and degree is nothing but that how many friends you are. So how much popular you are, that is your friendship network. Okay, now that is your degree. And then another very key concept of network science, which is also there in biological and mitochondrial networks, all these networks, is betweenness centrality. So betweenness centrality is how much central you are. It means even though you may have less number of connections, like right now, of course, if you have high degree, you have large number of friends, then you are very central. But sometimes it may be possible that you are between two large clusters. So if you want to go from one cluster to another cluster, you are in between. So you may have less number of friends, but if you want to go from, suppose, this direction to this direction, you have to always pass through this node. So it may be possible that you have a less degree, less friends, but you are still very central. So you are holding the network. So I am slowly coming to this holistic approach. So if we are having high degree, it means if I remove you, entire network will be broken down. Okay. And similarly, between a centrality, so if you are a very key player, even though you have less number of friends, but if you are connecting two communities, then if I remove you, your system is failure. So your system fails, correct? Similarly, I have another very key concept, which is clustering coefficient, and which is nothing but if your friends are friends too. So two people, if you are friends with these two people, and if they are also friends with each other, then your clustering coefficient is high. And diameter is how far you are from everyone. So very popular language, meaning these technical terms they have. So diameter is if you are measuring your distance with each and everyone in the system, and this largest possible distance is the diameter of the system, diameter of the network. And with that, there have been various different network models which have come into existence in last 20 years, though this network field started in 1950s by two Hungarian mathematicians, Erdos and Reni. Out of the mathematical curiosity, they wrote several series of papers because they found very nice phase transitions and phenomena in a very simple way of construction of the network. So that was in 1950s. And recently, like 20 years back, a group of computer scientists led by physicist Albert Barabasi, he found that he did a lot of different experiments, and he found that almost all the real world networks, including your metabolic networks, and other biological networks, and World Wide Web, which is technological network, and then social networks, all follow the same principle. And that principle is scale free. So they are not random. So he defies this concept that real world systems are not random. They have some properties, and which is popularly known as scale free property. And in popular terminology, if I say that few nodes have very large number of connection, and those connections are as large that these few nodes that carry entire network. So you can immediately think about the benefit and drawback of such kind of system. What are the benefits? Benefits are for such kind of systems where few nodes which carry the entire system is that if you remove all these peripheral nodes, or 90% of the nodes if you remove from the system, is still you don't break the network structure. Okay, so if you keep on removing the links which is not involving this node, then your system is still intact. And what is the drawback? If you have targeted attack, it means if you remove these hub nodes, your system gets collapsed. So such kind of system, natural systems, hence they are very robust against the random removal of the nodes and links, but they are very fragile or very sensitive to the targeted attack. So real world system, you can see that they are very robust. You have low node link deletion all the time. It is happening in the biological and real world networks, but they are very robust. And one of the key mechanism behind this robustness is achieved by that they are scale free. It means few nodes, so they have the very large number of connections. And there is another popular network model, which is a small word network, which is also given by during the same time in 1990s, like around 20 years back, by two physicists, Stogatz and Watts. And they showed that all these real world systems are very small. Small, it means the diameter is very small. And the popular language, we say that, oh, it's a small word. Everyone is connected with everyone in few steps. So it does not mean that you have many links. It's just that network organization in such a manner that you can go from anywhere to anywhere, only in few steps. Like one of the popular example which I can give, suppose you want to go from one very small town in India to a small town in US. So how many flights you have to take? Barely 3 or 4, because from your small town, you go to Delhi. From Delhi, you will go to New York, for example, and from New York, you will get flight to the that small town. So even though if you want to travel an entire globe, in very few connection, you can reach everywhere. So this is the popular transport example I have given, but that is existence everywhere in the nature. So any biological system, technological system, any system you take, it's almost a small word. So you from one place to another place, or from one node to another node, you can reach in very few steps. And that is known as a small word network. So there are 3 most popular models in the network science, which I have already introduced to you. And then there are several variations, which I am not bothering about. So Erdos-Reni random network, everything is connected with everyone randomly. That shows a lot of beautiful properties. But it turns out that real world systems, they have a much more richer structure, namely scale free, few nodes are hub, and a small word, it means a small diameter. Now, why we should investigate networks? So I have introduced the network. I have said that we live in the world of networks. I have introduced few popular models, but why we should investigate network? Now, this is the question which we can ask here. So I have written here few examples of or few applications of this network investigating a complex system or studying a complex system under this network science. Okay. So one is that if you want to understand the vulnerability of a system, if you want to understand the structural organization or functional behavior of large scale complex system, or temporal behavior, that how in times, like given this time, if you know the behavior, what would be the behavior at a subsequent times. So if you want to understand the properties of real world system, it has been shown that if we write down or investigate those systems under network framework, and network framework, you understand, we have only nodes and connection, and we lose all other properties. We are not worried about the size of airport, for example. We are only taking one information. So it is very reductionist approach, which we physicists often adopt. So it's a very reductionist approach. We only take airport as a node, okay? And if two airports are connected with a flight route, we make a connection. So here we are not talking about which airlines they are connected with, what is the size of the airport, all those information we are not talking about. Similarly, in the biological network, different chemicals may be, so chemicals are the nodes, irrespective of what those chemicals correspond to. And if one chemical is interacting with another chemical, you make a connection. So it is very reductionist approach, but still, despite of such a huge reduction, we still get many different properties. We can understand the mechanism behind the lot of behavior depicted by this real world system. For example, in the brain, you can think brain as terms of neuron and axons. So neurons, they are firing, and if one neuron, it is affecting another neuron through axon, then we make connections. So now your entire brain can have underlying network structure, and defying all other, like forgetting about all other properties of the brain, we can still investigate a lot of different mechanism, lot of different behavior of the brain using this network framework. So now, as I had already said, that a lot of different systems, real world systems, are scale free, and biological networks are also scale free. So what does it mean? It means that few proteins, suppose this is a protein interaction network, okay? So one protein is interacting with another protein through the functional performance or through the structural transformation. So few proteins, they act as hub. It means they interact with many other proteins, okay? And these proteins, they control the major functional aspect of cellular machinery, and they make the network robust and specific to attack. So whatever I have just said, and it's written here in terms of the biological networks. So your proteins, few proteins are only the hubs, and they are interacting with almost all other proteins, and they govern or they are key behind many cellular machinery. Metabolic networks have a small word topology. It means, and these are all research articles, which you can see the references here. So they have been investigated, and it was found that metabolic networks have a small word property. It means they have very shorter path lengths than the corresponding random networks. So if we connect the things randomly, then they will have a bit of larger path length, but they have much shorter path length than the corresponding random network. And it indicates that local perturbation in metabolite concentration could reach the whole network very quickly. So this is the implication of these small word properties of these networks. And another concept, like which is I had already introduced, clustering coefficient. Okay, so this biological networks, they have been found to have very high clustering coefficient, and which is directly related with modularity. And modularity, which we have tried to put here using this diagram, you can see they are modular, they're hierarchical structures. So here they are forming some subgroup, and this subgroup is interacting with this bigger subgroup. So this kind of hierarchical structure that have been found in a lot of biological networks, and these are the research article from where you can get all the details. But the key concept is that real world biological systems are scale free, they are having a small diameter, and they have very high clustering coefficient, or they are modular, they have hierarchical structure. So these are the basic properties which have been found in biological systems. And here, if there are questions in the question and answer box or chat box, I cannot see any questions. But if there are questions in the box, please, moderator, if you can read those questions. Are there any questions? So I don't see any questions in the chat box, but question and answer session, yes. Yeah. So now let me see the questions. So Hi, Sandra. So you have a question, how to identify a biological network under study is scale-free or a small word? Okay, very good question. So that's why I have introduced few technological terms, degree. So if we have a biological network, so first you have to identify what are the nodes, what are the connections. So any system which is given to you, first thing is that you have to identify what are the nodes, what are the links. And you know, for one system, you can have different types of the network. For example, let me now start with one example. Suppose I am talking about, suppose I am giving you scientists' name and their papers. And I'm seeing this complex system, what could be the nodes and what could be the connections? Okay. So you can say that nodes could be the scientist. And if two scientists, they have written paper together, you make a link. Okay. This is one way of defining the network. Another way of defining the network could be what? Anyone has any idea? So I can give some idea and then you can think later. So if one scientist has cited anyone else's work, okay, then here your papers are the notes. So, you know, not scientists, papers are the notes. And if one paper cites another paper, you can put a link. So same system, same data, information is same. But depending upon what properties you're looking at and based on how different ways you can think of the nodes and links, you can define various different types of the network given the same data, okay? So now to your question. So biological networks, first thing is that what is a system? You have to define the nodes and link. Suppose your biological system is metabolites, okay? You want to understand the metabolism, okay? Then your metabolites are the nodes. And if one metabolite is interacting with another metabolites or functionally they are interacting, then you can put a link, okay? So once you know the link, what you have to do, link is nothing but a degree. You have to calculate the degree of each node, and degree is nothing but the number of connection each node is having. Just calculate the degree and plot the distribution of those degree. If distribution is coming straight line in the log scale, and technical details are there in my paper, or you can try to read those paper. They are very elementary paper, the one paper which I have told in the physics news. So you just plot the log plot in the so x-axis and y-axis. So x-axis you put log k is the degree, and y-axis log of rho k, and it's a straight line. It's a scale free, okay. And a small word, you have to calculate the diameter. It means, again, there are software which are available. And I will also show you the one GitHub library where we have put all the software. You can go to that GitHub library and you can find out those software. In fact, in MATLAB, all this calculation of whether the network is scale free or a small word, those software, those codes are already there. So in MATLAB, you can just give one command and it will tell you whether the network is scale free or what is the diameter. So if diameter is smaller, then you can say that it is a small word. And diameter is small means that as you increase, so diameter should be approximately of the order of log n. So if n is the system size, n is the number of nodes, then diameter, if it is approximately log n, then you can say that this is a small word kind of network. So this is your first question. And there are techniques and all the codes easily available in MATLAB and in Python, which can calculate all the network's property. But key thing here is that the data, the biological data which you are taking and how you are constructing the network. Once network is constructed, then the problem is very easy. Now, another question by Vishal. As a person who is interested in amalgamation of machine learning and microbiology, sharing as a recent graduate, how should I go about learning and applying machine learning to microbiology? Okay, so I have not started this aspect. So I have so far not applied machine learning to biological system, but machine learning, I have recently started applying to the dynamical systems. Okay, but machine learning has some, I will show towards the end of my talk, some of the applications which consist of like machine learning predictions of the drugs, for example. So how employing machine learning techniques, you can predict the novel drugs. So this is one of the application which I have immediately in my mind. Second application, which is a very celebrated problem, like for example, can you predict the onset of epileptic seizure? So we are given the time series, and as you know, that you cannot predict the onset of the seizure. Okay, sudden there is a person start showing this seizure symptoms, and this time series, if you are measuring the person's time series in very short interval, only time series shows some change which can predict the seizure. So the thing is that how one can employ machine learning techniques in order to predict the occurrence of seizure. So there must be some changes in the time series or something, but how machine learning technique you can employ to use that predict the seizure. So two examples which I have in my mind, and as it comes, I will share with you all. What are the software to build networks? So in the Python, you have the software and codes, I don't remember, but in the GitHub library, we have shared everything. So Python and MATLAB are, as far as I know from my research lab, because I use Photon, still I am using Photon, but there are already inbuilt codes on software which can, you give the number of nodes, you give the number of connection which you want, and they can build the networks, okay? But from the real data, if your question is that from real data, how to build the networks, then from real data, you have to first take the data and you have to then see that you have to define the rules and then you have to build up the networks, okay? So if a new molecule is identified later on after the discovery of, okay, actually this gave me a very interesting example also. So right now he's telling that, suppose now we have built up the network and then a new molecule is identifying later and then new pathway. Then we have to keep on adding. This is the first thing, that we keep on updating the database. But second thing is, which is a very important hot area of research, which is the prediction of missing links. So it means you have some available data, okay? You have constructed a biological network based on available data. And then there may be some missing links, which is not there, is still visible. We don't have enough data. We have only one small segment of the data. So how one can predict the missing links? And there are many traditional ways to predict the missing links based on many statistical technique. But the recent interest which lies using machine learning concept to predict the missing links. This is exactly the question which is raised by, I don't see the name here, but this is exactly the question which is raised by him or her, that with the limited information, can we predict the information which we don't have because of the experimental barrier or because of the discovery of like experimental barrier actually, or some changes which are there in the system, can we predict? Given the network at this time step, so there are two things, missing link prediction. And second thing is that suppose network is evolving in time. So suppose if I am giving the evolution of the network until certain time series. So at time t0, I know the network. At after 1 year, I know the network. After 2 years, I know the network. Can we predict the network on later on time? So t0, t1, t2, by doing the experimental methods, I know the networks. But can I predict using the past information? Can I predict the temporal network? So this is also very hot area of research, and there's a lot of scope of machine learning, which is there in these two problems. Since now few papers report exception to scale-free nature of real-world networks, yes, or should explain the non. Yes, so scale-free was a very big success in 1998 and 1999. Like almost it gave rebirth to this graph theory or network science field. And it shows that almost all systems, they have a few nodes making up, but there could be exception of this scale-free network. I cannot recall any example right now, but there could be exceptions. But the key point actually, which I would like to emphasize, is that the real world systems are not random. So Baravasi, when he was leading this computer scientist, they had this thing in their mind that before that, the theory was that everything is interacting with everyone and randomly. So if you plot the degree distribution, it should be Gaussian. So they tried to extract the real world data, and they just tried to see by just assuming that will show the Gaussian kind of distribution, which we feel for almost all the aspect for the complex system. But surprisingly, they found that they are not random. So this is the first key discovery. And then they found, okay, there is scale-free, small word, everything. But the key point here is that they are not random. So things are not interacting with each other randomly. So they have some mechanism or some driving force which is responsible for the evolution of the complex system which we see today. So things are not interacting randomly. This is the key point. With this, okay, let me proceed, and please keep on putting the questions here, and I will again take up them in some time. I have to, okay, I have to, sorry, I want to close this session of question answer. It is popping up here, but I don't know how to close it down. Can anyone help me with that? I cannot close it down.
Yeah, just 1 minute. Just 1 minute. Are you able to?
Yeah.
Yeah.
Because that is popping up here, question answer list, and I have to close them down so that I can see my slides and proceed.
You can also minimize that.
Yeah, I think minimize. Yeah, that's what I'm trying to do. I am So maybe I can skip the slide mode and then, yes, it's done.
Yeah, can you just
Escape button, that works. Okay. So network science and medicine. So network has been the graph theory or network concept, which is a holistic concept. So network concept is that you don't treat the pathways individually. So one pathway or one node on its own may not be important, but that may be playing a very strategic role in the entire system. So network field is more like the holistic approach. So this has given a new view to the medicine, that is that instead of looking at one disease, for example, or few localized things, you see that how much it is important in the network structure. And diseases, how they are linked with each other. Can you have one drug to many medicine kind of concept? So all this concept that has their platform lying at the graph theory field, which is the holistic approach, where your position is much more important than your own property. How you are connected with everyone, that is the essence of entire network field, not your own chemical property or physical property, no, where you are placed in the graph. So this structural organization is the key of this network field, and that has given a new way to design medicine. So identification of disease genes and disease pathways. So pathways, and this is very important thing, and that has been used for, so there are several references which I have written. So you can see this Baravasi, he has also very much involved in applying this network science concept in various kind of diseases. And there are several investigations which have been done on the disease network. So disease network also have certain kind of network architecture, which has been emphasized, and very sophisticated technique of random matrix theory. So random matrix theory was in the beginning, that was introduced in order to understand the complex nucleon interactions, so electron, proton, and nucleon interaction in atoms. So that was the basic background of the random matrix theory. And recently it has been shown on the network field in order to find out the important proteins or important nodes. So that has also been shown that disease network have lower randomness as compared to the healthy network. So these are the fundamental curiosity or fundamental level, but it has application in the drug development and the drug target developments. So this random matrix theory paper is the last one, which is from my group. And the very recent hot area of research is aging, in order to understand that what drives our aging. And aging, like as network scientists or physicists, they understand, or mathematicians understand, and aging is a complex phenomena involving multiple biological processes. And there have been investigation or efforts to understand aging in terms of graph theory or networks. And the networks and machine learning, the question which was asked, that I have never worked on so far biological network or machine learning. So far, we have started working on the machine learning and dynamical system, but of course on this missing link kind of thing. But I have so far not applied this problem to the real world biological system. But using machine learning, can we identify keynotes in the network? Can we predict the missing link and the information which I have just given? Can we predict the evolution of the network or network classification with limited time series information and few nodes? And some of the references which are listed here, some of them are from my group and some of them are popular cited articles from other groups. But I'm afraid here so far not much, which at least I am aware of in the particularly to the medicine or biological system, machine learning, except those things which I have just quoted, which is of the general curiosity, that missing link information, or false positive links, some links which are present, which we show by which we extract from the experimental data, but they are not there in the real system. So those kind of, can you know that which are the false link or which are the missing links? So those things at the fundamental level people have done, but using machine learning, there are only recent attempts in this direction as far as I know. Networks and big data. So, as soon as you have biological system, you always have big data, like in genomics, which I will come also. I have a few slides on the genomic, particularly on the genomic things. So all this concept which I am telling here, they are also applicable to all the biological networks, including the genomic networks. So you have big data, multi-omics data from genome, metabolome, protein. And then the question is that, can we define the biomarker or disease subtypes? Can we have some the drugs which can cure for some disease? And For any further knowledge in big data, I had my PhD student who is currently in San Diego, and email ID is here. So anyone wants to know the direct application of this multi-omics data, big data, and network construction, please feel free to write a mail to Pramod Shinde, and he'll be happy to. Actually, he has only provided me this slide to show, and he will be happy to help you all. So now let's come to this nucleotide co-occurrence network. So I have only few slides on that. I will be very happy to address the questions if you have. I will keep on stopping in between, and I will keep on taking the questions. How much time I have? Okay. So genetic variation impart their effect as a coherent. Manifestation of complex disease also depends upon the interaction of variable sites. So these are only the gist. And in the last slide, let me first talk about how we are constructing this genomic networks. So human mitochondrial genome assembly has this many base pairs, okay? And then using this many base pair, how we are constructing the genomic co-occurrence network, okay? So here the important thing is co-occurrence. So how one mutation co-occur with another mutation. So as soon as I am saying co-occurrence, I am again talking about the links, okay? So two things co-occur together, and this would be here basis to form the link, because as I said, then when we talk about the networks, we need to know what are the nodes and what are the links. So in the genomes, I am putting nodes as a sites, okay, variable sites, and the two variable sites, they are linking if they are mutating together. So if co-mutation, so I have defined the co-mutation, and in that basis, I have constructed the co-mutation network or co-occurrence network. So why human mitochondrial one is taking? Because it has it doesn't change with the evolution, and it preserves a lot of properties which one can see during the evolution, and hence mitochondrial genome sequences have been investigated, and that's why we are also interested in investigating mitochondrial genomic sequences. So there are two different ways one can construct the network: co-occurrence network, co-mutation network. And I will tell you that the same technique which we use to construct this co-occurrence network for mitochondria, same techniques can be used to construct the correlation network using the financial time series. Financial time series means you have the data for share market, and then you can construct the correlation matrix using almost the similar concept which I am going to use here to construct the correlation matrix using the share market time series. So co-occurrence network, all the links, all the codes, etc., it is there in this GitHub repository. So you guys can download, anyone can download, and this is a README file, and very easy to use. These codes are there. So you put your data and all the information that is there in this GitHub library of the complex system, and you can see this thing. So codes are there. So now suppose if we have all the variable sites from different samples, okay, so you have different samples, and this is a mitochondrial sequence, okay, and one only takes the variable site. So here you see there is no variation here, there's no mutation here. Variable sites means the site which has undergone at least one mutation, okay, that one is taking. So this is different samples, and those sites which are not mutating, one is not taking those sites, but only those sites which are mutating, we are taking only those sites for construction of co-mutation network or co-occurrence network, okay. And then one defines a very, I promise that I will not show any mathematical equations, but this is a very preliminary equation which shows the correlation between the ith and jth unit. And similar kind of concept one can use for financial time series. So Cij is the co-occurrence frequency between I and j variable sites, and Cij would be between -1 to +1, or it would be between -1 to +1, or you can take the mod and it is between 0 to 1. So 0 means there is no correlation between two sites I and j, and 1 means this is perfectly co-mutating, correct? And by putting the different threshold, one can construct the different networks. So now after putting this co-occurrence frequency and all the codes to construct the correlation network is already there in the GitHub library. And all the detailed information is there in this paper, in this publication from our lab. And with that, one construct the actually correlation or co-occurrence network. So this is perfect correlation. Yes, this is not perfect co-occurrence. Okay. So one construct this kind of thing. And this is some working example, but I'm skipping that. And I would advise you to see this paper where also we have given this working example. But the key here is that if two variable sites, they have their mutation are happening at the similar time. So if they are mutating at the same number of times, so same sequence, if suppose this site is mutating and this site is mutating in the same sample and this frequency is higher, then you have more relation with the co-mutation. So it means two variable sites are mutating together. So it depends upon how many times in how many samples this two sites are mutating together, and that describes the co-mutation. And using this kind of network construction, we try to understand the properties of evolution and everything. And this is the network which one can see with the co-occurrence threshold. So if co-occurrence threshold is 1, then only those sites which are perfectly co-mutated, that would be connected and all other would be gone. And if co-occurrence threshold is 0, then you will find that everyone is connected to everyone, okay? Which is not very interesting case. This also here you can see the right-hand side, extreme right-hand side. Now this is also not very interesting case because you have here very disconnected, disjoint network. So you don't have very rich, interesting network pattern. You have only modules, which are also important, but we are most interested in some kind of complex interaction pattern. And now, as I promised you that the same kind of techniques, the correlation kind of techniques with variation, but is used in finance network also. Okay, and in the finance network, this kind of correlation matrices people are using in order to understand the risk involved or influence of corporates, financial security and stability, quantitative analysis of economy. So all those kind of things has been done using co-occurrence or correlation network in this paper. And similar techniques we have used for the genomic network. So, same technique, exactly the same technique, random matrix approach, all these two different techniques. Same technique can be used in two different systems. One is financial network and one is this genomic network, which is also from our lab. And in the same direction, we can construct the co-mutation network, which let me take now some questions and then I can proceed further. Now, actually, it's difficult. Okay. So questions are since now few paper. Okay. So Aishwarya has a question. Depending upon mitochondrial DNA mutation network produced, can we predict the viability of the mitochondria? Is yes, how can we do it? So can you elaborate your question, Aishwarya, a bit? Maybe I am not able to understand your question. So maybe if you try to elaborate your question now, or maybe we can talk to you. So I can tell you that what we have done so far, because this is a very new area of research which we have involved in. So using this mitochondrial DNA mutation network, we have found few modules which are very important. So now we have applied this kind of technique into different population lying at a different altitude. Okay, and we have tried to find out that what makes a population habitable to live in a high altitude. So those kind of investigation we have done. And we have also taken the mitochondrial genomic sequence for the different geographical region. And we have tried to see that at the mitochondrial level, which geographical sequences or which geographical data, they are matching to each other. They are closely matching to each other. What are the signatures for different geographical locations? And so far we have not applied this thing for any disease kind of network, but that is our aim. But like different altitude population, for example, Tibetan population, how they are living, what mitochondrial signature which is there in the Tibetan population, than those which are there at the lower altitude. So this kind of things which we have tried to address using this example, using this kind of techniques. Is there any other question? If not, okay, let me proceed. So in the similar manner, we can also do the co-mutation, which is a similar technique, and all the detailed information is there in these two publications, which you can refer from our lab. And there are some other labs also which have used this co-evolution mutation or this mitochondrial genomic sequence in order to understand the biology behind the system. And with that, I would also like to introduce to a very important aspect of motifs. So motifs, as shown by Yuri Allen in Nature 2007 paper, that motifs are nothing but the this complete subgraph. You see, this is a complete subgraph. This is 2-order motif, and this is 3-order motifs. And he emphasized that in the biological system, these motifs are the building block of any biological system. And in the evolution, these motifs are preserved. So that's why we have concentrated on these motifs, and we have tried to see that which kind of motifs are found and which geographical locations and different systems, what kind of motifs structure which we have. So statistics of the motifs structure that we have used. And there's one more kind of motifs, the feed-forward motifs, which people, biologists, have been a lot interested in. So different application, identification of ancestral modules in human population, that has been done using this kind of analysis of the motifs, and motifs is nothing but the complete subgraph. So putting the different threshold, one can see that how motifs are evolving in time and can try to analyze them. With that, let me take one more question. Can you please comment on how we can decide the threshold value? Yeah, this is a very, very interesting question, that what is the threshold? So I have tried to ask these questions by biologists whom I had collaborated with and I have been collaborating with. So there is no answer, no exact answer that which co-occurrence value, which threshold value. So identification of threshold is an open problem. So mathematically, we have tried to find out the threshold. So we take threshold. So it's a very good question in terms of technique, employment of the technique as well. So what we did actually, so suppose if we make threshold 0, then everyone is connected to everyone. We are not interested in that. If we take threshold very high, then what will happen? You have this segmented cluster. So what we did, we kept on increasing the threshold, and we see the moment my network start breaking into the different cluster, we stop at that point. So what happens that we have identified the threshold in such a manner that it has all the nodes, almost all the nodes inside the network, and as less connection as it can have. So that will give us some meaningful network which we can analyze to. So this is the one way of finding the threshold. Another way of finding the threshold is that you keep on increasing the threshold, and if you see that some drastic change is there occurring in the network, that also you can take as a threshold. But one very safe way is that threshold should be in such a manner that you don't get disconnected nodes. So as soon as you start getting disconnected nodes, you stop there. So this could be the one way of finding the threshold. But as far as biological thing, no, there is no straightforward answer to how to find out the threshold. So with that, actually, let me go towards the end of my talk. And because these are some results which I think if you are interested in learning this field, you can read some of these papers and then you can write me back and I will be happy to help, or anyone in my lab or the authors of this paper who are from my lab. If you can write down Rahul, who is now key expert in mitochondrial motifs, and Pramod, who has already graduated, they will be very happy to help with you. But here take-home message is that motifs are important because they are complete subgraph, which are known to be as a building block of any complex system, and they are preserved during the evolution. And using this co-occurrence technique, first we constructed the network, okay? And after this network, we have tried to identify the motifs, okay? And using these motifs, like this example is for the different altitudes. So we have constructed the network at the different altitudes. So this altitude, we constructed one network, this altitude, one network. So all this altitude, we constructed one network. Or if we have, so we have different ways of constructing the network. Either we can construct one network, combining all the sequences, or depending upon if we have many sequences here, we can also construct the individual network for one altitude. And then we have tried to analyze the motif structure and try to distinguish this altitude based on the motif structure with the information which is there in the mitochondrial nucleotide. So mitochondrial, which is preserved during the evolution. So then we expect to get some information about the evolution using this motif structure. With that, there's a conclusion that network science provide. So this is the take-home message is that network science, it provides the powerful framework to identify the mutual role of nucleotides. Networks of nucleotides and genes, they uncover or they emphasize the role and importance of the interaction in context of human population. And such kind of technique can be extended to analyze the patterns in the disease. Again, like if you compare the disease networks and the normal healthy person network, and then again analyze this motif structure, which is constructed after, which we get after putting the threshold. Then what information we get, or what kind of different statistics we find among this motifs, motif statistics. And the one problem which we are also interested in, and this is a very hot area, is aging. So can we also try to understand, using this mitochondrial genomic sequence of young age and older age, can we understand this phenomena of aging? With this, let me now acknowledge DST, CSR, and BRNS for funding and financial support. This is my complex system lab. You can find almost all the papers here at my website, and as well as a list of complete papers on the biological section in this Google Scholar. This is my bio module, like it means all these 4 students, they have already, so 3 of them have already graduated and postdoctored at different places. They have done all the biological network things in our lab. And before I take up the questions, let me also share some internship opportunities, because yesterday Gayatri, she told me that I should also offer some insight to the future perspective to you all. So in research wise, of course, it's a very hot area of research, this biological networks, but there's some internship opportunities which is provided by IAS, Indian Academy of Science. And the entire this procedure is written here. And Sanidhi, who is doing her internship in my lab through this IAS fellowship, she has kindly agreed to address any concern which you will have later on to apply this fellowship. So her email ID is here, and you can talk to her later of all the entire procedure, and they select and they give you some fellowship. And using that fellowship, you can do internship in any of the lab in India, not in my lab. So any biological lab, then you can do the internship and learn more about the subject. And there are some industrial application. Again, as Gayatri asked me to emphasize, I have never done an industrial application, but one of my graduate student, Aparna, she has opened some company, and she is the CEO of that company. And she actually, you can contact her more, but see here there are certain applications, industrial application, which is written in this slide. And for more information on this, you can directly contact with Aparna Rai. She is also, again, kindly agreed to help you guys as much as she can. With this, thank you so much for your kind attention, and that's it. So any question, if there are further questions, I'd be happy to take up those questions.
Thank you, Dr. Jalan, for a very interesting talk on the mitochondrial networks. So there was a few questions. You already took the questions during your talk, which is very nice. So what I'll start with one of the questions that is left out. This is a question by Bipin. He's asking if, since now there are few papers that report on exception to scale-free nature of real networks, how one should explain the non-scale-free nature of real networks?
So I think I addressed that question, that you have, there may be some real world systems which are not scale-free. And I think you want to ask the question that how do we know that the system is scale-free or not? So there are software, there are codes which you have to only plot the degree distribution. In case you don't know how to plot, there are already some software which is there in MATLAB and Python, which can immediately, you give the network and they will tell you whether it's a scale-free or not.
Okay. All right. I have one question.
Please, Neha.
Disease networks show sort of dissociative degree of correlation. What do you mean by that?
Okay, dissociative and associative is a very useful concept, which I just like. Associative means high degree nodes if they are connected with high degree nodes. Then those kind of networks are associative. And dissociative means high degree nodes are connected with lower degree of the nodes which are having low degree. And biological networks, actually most of the real world networks have been like social networks, they have found to be associative. It means high degree is connected with high degree. But biological networks, I think they are mixed up. A lot of biological networks are disassociative. It means high degree nodes are connected with lower degree. So these are the observations, and one can ask what is the consequence of these observations. So that also shows a very basic nature of different systems. Like, social networks and biological, they both can be scale-free, for example. But social networks are mostly associative. High degree is connected with high degree, and biological network connected high degree with lower degree. So maybe that says that how we behave the things socially and how nature prefers us to do the function like at the biological level. Perhaps both can be scale free, but and then dissociative networks are hierarchical in nature. So that is also one of the consequence of being disassociative.
Okay.
So and okay, one more thing I can tell based on disassociative. You know that it has been emphasized that in the evolution, first things duplicate. So duplicate, and then after duplication, it changes performance. So you will find this duplicate structure. So two nodes having the same kind of link, same kind of interaction pattern in the network. And that also is one of the reasons that why biological network of disassociative, because that leads to disassociativeness. So two nodes having exactly the same interaction pattern, almost the same interaction pattern. And that is also one of the key behind the evolution, which has been emphasized. Duplication. So each gene first duplicate, and then it changes its performance so that system remains robust. And that property also leads to, can lead to disassociativeness in biological systems.
Oh, yeah, thank you very much. It was a very interesting talk, and thank you for enlightening the audience.
And if anyone has any
questions, I can take one more question perhaps, because we will run out of time soon. Okay, there is.
I can read this question in question answer session. I think.
So
I deeply desire to study mitochondrial network. I would love to do an internship in your lab. Is that possible? You can send a mail to me separately, and I'll see. My study area is cancer biology, and I specialize myself. Okay, that is the same thing. Okay, so you can write down later.
So perhaps what we can do is direct them towards the different opportunities and on the mitochondrial networks, and anyway, in case all the feedback forms will be sent to you, so and the questions as well. So if you want to direct them towards the right studies, then that would be great. So before we end, I have a couple of announcements to all the listeners. A feedback link will be sent to all the participants, on completion of which you can receive your participation certificate. Also, all feedback forms will be forwarded to Dr. Jalan, as mentioned earlier. I hope, the next webinar will be on 10th of September. It will be by Dr. Richa Singh from IIT Jodhpur. Also, please follow us on all social media handles for updates and information on the webinars. A recording of this webinar will also be available on the Manav YouTube channel. So thank you again. Thank you, Dr. Jalan, for taking your time and very enlightening us with all the complex network systems. So thank you very much. And
thank you.
Thank you all the participants for attending this webinar. Thank you very much.